Hello students, myself Bhanu Solanki, Senior Faculty Sri Chaitanya, Institute of Education. Today we are going to discuss one more chapter, that is Biotechnology Principle and Procedure, and its application part also. Very significant chapter from exam point of view. Here we must remember that biotechnology means first its introduction and definition part is here means technology using live organism or their enzymes to produce the products and processes useful for the human being. Product and services by using organism or their enzymes or their components, that it is. In this case, if we consider, then the biotechnology can be divided as a classical, that includes curd formation, beard, wine, etc. bread also, that comes into the classical. But if we think about the modern biotechnology, that means this definition is only just in a restricted sense. The process which uses genetically modified organism to achieve the same on a larger scale, that is the modern biotechnology. Means what comes under this process? Look, in vitro fertilization to produce test tube baby comes in this part. Synthesizing a gene and using it, developing a DNA vaccine, correcting a defective gene means gene therapy, they all comes into the modern biotechnology. Then the definition suggested by European Federation is also given here. According to European Federation of Biotechnology, biotechnology that encompasses both traditional view and the modern molecular biotechnological view. What it is? Integration of the natural science and the organism and their cells and part and the molecular analogues to produce the product and services of the human interest. That is the European Federation definition. Principle and biotechnology. Development of the modern biotechnology was based on the two core techniques. Number one, genetic engineering, means that technique that alter to the chemistry of the genetic material to introduce these into the host organism and thus change to the phenotype of the organism. Second, bioprocessing engineering, means maintenance of the sterile, yani free from contamination, ambience into the chemical engineering processes to enable the growth of only desired microbes, cells in the large quantities so that the desired product can be obtained without any kind of contamination. So these are the two core techniques because of which modern biotechnology came in existence. Here they mention advantage of sexual reproduction over the asexual reproduction. We know that it provides opportunities for the variations and the formulation of unique combinations of the genetic setup, which may be beneficial for the organism as well as for the population. Asexual reproduction preserves the genetic information, means whatever the genetic constitution we achieve, that would be keep maintained here. While sexual reproduction permits variations, but we know that traditional hybridization processes used the plant and animal breeding very often lead to the inclusion and multiplication of the undesired genes also. Means one of the disadvantages of the sexual reproduction is what? That beside a desired gene, some undesired gene will also be incorporated, and that's the point of challenge. Consequently, the recombinant DNA technology, yani genetic engineering, which include creation of the recombinant DNA, use gene cloning and gene transfer, overcome this limitation and allow us to isolate and introduce only a set of desired gene without introducing any kind of undesired gene into the target organism. This is the biggest achievement or advantage. Do you know likely the fate of this piece of DNA which is somehow transferred into an alien organism? Yes. So this is because the alien piece of the DNA has become a part of chromosome which has the ability to replicate itself. In a chromosome, there is a specific DNA sequence that is called origin of replication initiation, which is responsible for the initiating to the replication. Therefore, the multiplication of any alien DNA in an organism needs to be a part of the chromosome which has a specific sequence known as origin of replication initiation. Multiplication of the gene or the DNA is known as gene cloning. Multiplication of the desired DNA containing cell is the cell cloning. Let us take an example. first instance of the construction of artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of the linking of the gene encoding antibiotic resistance, linking of a gene encoding to the antibiotic resistance with a native plasmid of Salmonella typhimurium. Stanley Cohen and the Howard Boyer, two scientists, accomplished this in 1972 by isolating antibiotic resistant gene by cutting out a piece of DNA from the plasmid which was responsible for conferring antibiotic resistance. The cutting of the DNA at the specific location became possible with the discovery of so-called molecular seizures, means restriction endonuclease enzymes. The cut piece of the DNA was then linked with the plasmid DNA. These plasmids DNA act as a vectors to the transfer of the foreign DNA or the piece of the DNA attached to it, and then this you know that that particular linking of the antibiotic resistant gene with the plasmid vector become possible with the enzyme DNA ligase, cutting by restriction endonuclease and joining by DNA ligase. Now these recombinant plasmids were, when this DNA was transferred into the E. coli, a bacterium closely related to the Salmonella could replicate it using this new DNA of the host and polymerase or DNA polymerase enzyme and make the multiple copies of it, and they also become resistant for the antibiotics. So antibiotic resistant gene was isolated from the one bacteria and transfer into the plasmid, so plasmid become recombinant, and then these recombinant DNA plasmids were introduced into the other organism. Means it was one of the case where first time genetic engineering lead to the transfer of the antibiotic resistance into the sensitive bacteria. You can hence infer that there are three basic steps. Number one, identification of the DNA with the desired genes. Number two, introduction of the identified DNA into the host. And number three, maintenance of the introduced DNA into the host and transfer of the DNA to its progeny. These are the three basic steps because in this chapter sequence related questions are very common. So all these basic steps and their sequences you have to keep in your memory. Tools of the recombinant DNA technology. Yes. Now we know from the foregoing discussion that the genetic engineering or the recombinant technology can be accomplished only if we have key tools. What they are? Restriction endonuclease enzyme, means molecular seizures, polymerase enzyme, ligase enzyme and vectors, and the host organism in which the DNA is to be transferred. First of all, restriction endonuclease. In 1963, two enzymes responsible for the restricting to the growth of bacteriophages in the E. coli were isolated. One of these added methyl group to the DNA while the other cut to the DNA. The one which add to the methyl group is known as modification enzyme, and the one which leads to the cutting, that is known as cleavage enzyme, means restriction modification system. Here you must remember why restriction endonuclease called the word restriction in the restriction endonuclease indicates what? Restricting to the growth of bacteriophage. Because these enzymes were naturally present into the bacteria so that these bacteria can be saved from the attack of bacteriophages, so the name arises restriction endonuclease. After 5 years of this, the first restriction endonuclease HIND2, whose functioning dependent on the specific DNA nucleotide sequence was isolated and characterized 5 years later, means in 1968. It was found that the HIND2 always cut DNA molecules at a particular point by recognizing to a particular specific sequence of 6 base pairs. This specific base sequence is known as recognition sequence. So every restriction endonuclease enzyme has their unique specific recognition sequence, and most of the time it is made up of 6 base pairs. The convention for the naming of these enzymes, what is this? First letter of the enzyme denote to the genus, second 2 letters represent to the name of the species. Okay, fourth letter represent to the strain, name of the strain where from it was isolated. Just for example, in echo R1, R represented to the derived from the strain, RY13 strain, and the Roman numbers represent to the order in which this enzyme was isolated from the strain of bacteria. So once again, in echo R1, E stands for Escherichia, CO stands for the coli, R stands for the RY13, the name of strain, and one Roman number indicate that from this strain it was first isolated enzyme. Restriction enzymes belong to a large class of nucleases enzyme, endonucleases. There are two types of nucleases, exonuclease which cut from the termine, endonuclease which cut into the middle portion or into the intercalary positions. Each restriction endonuclease function by inspecting to the each restriction endonuclease functions by inspecting the length of a DNA sequence. Once it finds this specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at a specific point in their sugar phosphate bond, means phosphodiester bond breakdown. The restriction endonuclease recognize a particular sequence. In this recognition sequence, they cut in between the two particular nitrogen bases by cleaving their phosphodiester bond. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence into the DNA. You understand the palindromic sequence? When any sequence, by keeping same reading frame, suppose 5' to 3', in left to right and in right to left, if are same, GAATTC, GAATTC, such type of sequences are known as palindromic sequences. So for the echo R1, the palindromic sequence is shown here, GAATTC. Must be careful about it. This is 5', this is 3', this is 5', this is 3'. So this enzyme is cutting in between G and A, Consequently, a strand that arises is partly overhanging single-stranded form. So when overhanging single-stranded form come very close, so it facilitates to this hydrogen bonding, and this hydrogen bonding help them to establish the phosphodiester bond in between the cleaved parts. Yes. Here they mention that GAATTC, and in the same way GAATTC. Restriction enzyme cut strands of the DNA in a little away from the center of the palindromic site, but between the same two bases. Here it was cutting between G and A, here also it is in between the G and A. These are the overhanging stretches called sticky ends. This leaves a single stranded portion, and these are the overhanging stretches called sticky ends. These sticky ends are named because they form hydrogen bonds with their complementary cut counterparts. This stickiness of the ends facilitate the action of enzyme DNA ligase. Very important part. Restriction endonucleases are used into the genetic engineering to form the recombinant DNA molecule. Again, here they show that this is the vector DNA, it is cut, and this is the desired DNA, it is also be cut, both with help of the ligase joined together, recombinant DNA form, transfer into the cell divide to our DNA, recombinant DNA also multiply. You may have realized that normally, unless one cut to the vector and source DNA with the same restriction endonuclease, the recombinant vector molecule cannot to be created. Means one of the condition is that the source DNA, if you are cutting by restriction endonuclease X, so vector should also be cut by the X, so that the similar kind of overhanging single stranded stretches, means sticky ends, can generate, so that the joining can be facilitated. Separation and isolation of the DNA fragments. The cutting of DNA by restriction endonuclear results into the fragments of the DNA. These fragments can be separated by a technique called gel electrophoresis. Why? Since DNA fragments are negatively charged molecules, if they can be separated by forcing them to move towards the anode under the electric field through a matrix or medium. Nowadays, the most common matrix that we use is the agarose, which is a natural polymer extracted from the seaweed. You heard the name Gracilaria and Ziladium. Be careful, seaweed, not seagrass. The DNA fragments separated resolve according to their size, very important line, according to their size through the sieving effect provided by the agarose gel. Resolution of the DNA means separation of the DNA fragment is determined by the size of the DNA fragments. Very, very important. Size of the fragments is the main determinant. Hence, the smaller the fragment, farther it will move, means more distance it will travel. So this is one of the presentation of gel electrophoresis. The separated DNA fragment can be visualized only after staining the DNA with ethidium bromide. Ethidium bromide you have to use to dye the DNA fragment, and later on it should be exposed to the UV radiation, means in visible light we cannot visualize the DNA. In the UV light, when they treated with the ethidium bromide, they would appear visible light, they would appear of bright orange colored, they would appear bright orange color bands. These separated bands of the DNA can cut out from the agarose gel. The separated bands of the DNA can cut out from the agarose gel and extracted from the gel pieces. This step is known as elution. The DNA fragment purified in this way are used into the construction of recombinant DNA. You know what are the second tool is a plasmids. Plasmids and bacteriophage have the ability to replicate within the bacterial cell independent of the control of chromosomal DNA. Bacteriophage, because of their high number per cell, have a very high copy number. Within the bacterial cell, some plasmid may have only 1 or 2 copies per cell, whereas other may have 15 to 100 copies per cell. Their numbers can go even higher if we are able to link an alien piece of the DNA with a bacteriophage or plasmid. We can multiply its number equal to their copy number. So, for an ideal vector, its copy number should be high, so that multiple copies of our desired DNA or the gene of interest we can create or we can develop. The following features that are required to facilitate cloning into a vector, these are the main features. Number 1, origin of replication initiation ORI. This is a sequence from where the replication starts, and any piece of the DNA when linked to this sequence can be made to replicate within the host cell. This sequence is also responsible for controlling to the copy number. Means two functions: initiation of the DNA replication and controlling to the copy number. Number 2, selectable marker. It should also be present. In addition to ORI, the vector requires a selectable marker which help into the identifying and eliminating known transformants and selectively permitting to the growth of only transformants. I must say, selectable marker helps to distinguish transformants from non-transformants as well as recombinants from the non-recombinants. What they may be? Transformation is a procedure through which a piece of DNA is introduced into the host bacterium. Normally, the genes coding for resistance to any antibiotics such as ampicillin, chloramphenicol, tetracycline, etc. are considered the useful selectable markers for the E. coli. Number 3, cloning sites, yani recognition sequence. In order to link alien DNA, the vector need to have very few, preferably single recognition sites. Means every type of recognition sequence must be present in a single copy, because if more than one copies are present, so your vector will cut into the many pieces and then their joining would become a troublesome situation or the cumbersome procedure. Preference of more than one recognition site within the vector will generate several fragments which will complicate to the gene cloning phenomena. Here their PBR 322 is mentioned, in which two selectable marker ampicillin resistant gene and tetracycline resistant gene are shown. In the ampicillin resistant gene there are two selectable markers, there are two restriction sites, PST1 and PV1. In the tetracycline resistant there are also two recognition sites, BEMH1 and SAL1. In the replication of plasmid region there is a one recognition sequence, PVU2. And apart from this, ECO R1, CLA1 and HINDI3 are also present in the ECO R1. ROP, replication of plasmid, codes for the protein that involved into the replication of the plasmid. Means ORI will start and ROP will proceed it. Yes, BAMH1 site of the tetracycline resistant gene in the vector PBR 322, we can take an example. The recombinant plasmid will lose if we are inserting our gene of interest here in the BAMH1. So it means the recombinant plasmid will lose the tetracycline resistance due to the insertional inactivation. But still can be selected out from the non-recombinant by plating the transformants on the ampicillin containing medium. Means simple, the procedure is that if any bacteria which are recombinant because of the BAMH1 site, so they are tetracycline sensitive now, tetracycline resistant finish. So any bacteria which is capable to grow on ampicillin but not capable to grow on tetracycline, it means what they are? They are recombinants. If any bacteria growing on both ampicillin and tetracycline, it means they are transformant but they are not recombinant because insertional inactivation not happened. So here they mention it in such a way. Selection of the recombinants due to the inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on the two plates having different antibiotics. So to overcome it, therefore, an alternative selectable marker have been developed which differentiate recombinant from the non-recombinant on the basis of their ability to produce the color in the presence of chromosonic substrate. Such example PVU8, where there we will insert our DNA in beta-galactosidase enzyme gene. If recombination happen, so beta-galactosidase will not work. If recombination not happen, so beta-galactosidase will work. If beta-galactosidase is working, so the substrate of the medium X-gal when utilized, product will arise which is of a blue color. If beta-galactosidase is not working, so color will not produce. So it means on the culture medium, if any bacteria is producing blue color colonies, so it means non-recombinants, and if they are producing colorless colonies, so it means they are recombinant cells. So this result into the inactivation of the gene by beta-galactosidase gene. This result into the inactivation of this gene for the synthesis of this enzyme, which is referred as the insertional inactivation. The presence of a chromosonic substrate gives blue colored colonies if the plasmid in the bacteria does not have an insert. Presence of the insert result into the insertional inactivation of the beta-galactosidase gene, and therefore the colonies do not produce any color and they are recognized or identified as the recombinant colonies. Vectors for the cloning genes into the plants and animals. For the plants and animals and from the bacteria to virus, which have known this for the ages, how to deliver the gene, here they mention. For example, Agrobacterium tumefaciens, a pathogen of the several dicot plants, is able to deliver a piece of DNA known as the tDNA to transform a normal plant cell into a tumor and direct these tumor cells to produce the chemical required by the pathogen. Similarly, retroviruses in animals have the ability to transform the normal cells into the cancerous cells. A better understanding of the art of delivering gene by the pathogen in their eukaryotic host has been generated. The knowledge to transform these tools of the pathogens into the useful vectors for delivering the genes into the or genes of the interest into the human. The tumor-inducing plasmid (TI) plasmid of the Agrobacterium tumefaciens has now been modified into the cloning vector, which is no more pathogenic, means it will not cause any disease. Pathogenic to the plant, but is still able to use the mechanism to deliver the gene of our interest into the variety of plant. Similarly, desired gene into the animals can also be transferred, means the retrovirus and the tumor-inducing plasmid of the agrobacterium is to be modified in such a manner that their disease-causing ability finish. Such a type of vectors are known as disarmed vector, competent host. Since a DNA is a hydrophilic molecule, it cannot pass through the cell membranes. In order to force bacteria to take the plasmid, the bacterial cell must be first made competent to take up this DNA. To make them competent, this is achieved or done by treating them with the specific concentration of divalent cations like calcium ions, such as the calcium, which increase the efficiency with which DNA enter into the bacterium through the pores in its wall. Recombinant DNA can then be forced into the such cells by incubating the cells with the recombinant DNA on ice, followed by placing them briefly on 42 degrees centigrade temperature heat shock and then putting back into the ice, means cold, heat, cold in the calcium. So this calcium and this heat shock, they will allow the transfer of this, enable the bacteria to take up the recombinant DNA. Beside this, some gene transfer methods, direct gene transfer methods are also mentioned. For example, microinjection. Microinjection is the direct DNA transfer in animal cells. Similarly, another method is suitable for the plant is the biolistic or gene gun method. So, gene gun method suitable for plants. Remember, microinjection for animal cells, biolistic or gene gun method for the plant cells. Recombinant DNA technology involves several steps. Very important paragraph. First, isolation of the DNA. Number 2, fragmentation of the DNA by restriction endonuclease. Number 3, isolation of the desired DNA fragment. Number 4, ligation of the DNA fragment into the vector. Number 5, transferring these recombinant DNA into the host and then culturing the host cell into the medium at the large scale and extraction of the desired product. These points you have to keep in your memory as such in sequence. I repeat, number 1, number 2, number 3, number 4, number 5. The nucleic acid is the genetic material of all organisms without an exception. In majority of organisms, this is deoxyribonucleic acid DNA. In order to cut DNA with the restriction enzyme, it is need to be in pure form, free from the other macromolecule. Since the DNA is enclosed within the membranes, we have to break the cell, open the DNA along with the other macromolecules such as the RNA and protein, polysaccharides and lipid. This can be achieved by treating the bacterial cells or the plant cells or animal cell with the enzyme. So if we want to isolate DNA from the bacteria, treat with the lysozyme. If we want to isolate DNA from the plant cells, treat them with cellulase. From the fungal cell, treat with the chitinase. In this manner, we will obtain DNA. Now this DNA would be treated with the RNase and protease. Consequently, the all surrounding RNA and protein will digest. The RNA can be removed by treatment with the ribonucleases, whereas the proteins can be removed by treatment with the proteases. Other molecules can be removed by appropriate treatment and purified DNA ultimately precipitated out after the addition of chilled ethanol. This precipitation of the DNA into the chilled ethanol is known as spooling. This is the spooling. Cutting of the DNA at specific locations. Restriction enzymes digestions are performed by incubating purified DNA molecule with the restriction enzyme at the normal condition for that specific enzyme. Agarose gel electrophoresis is employed to check the progression of restriction enzyme digestion, that it happened or not. DNA is negatively charged molecule, hence it will move towards the positively charged anodes. The process is repeated with the vector DNA also. The joining of the DNA involving several processes. After having cut the source DNA as well as the vector DNA with the same restriction endonuclease enzyme, they are allowed gene of interest from the source DNA will be cut and cut vector with the space are mixed with the ligase enzyme. This ligase will result into the preparation of recombinant DNA. Here their PCR, polymerase chain reaction, is also mentioned. Just keep the points. It is the process of in vitro DNA replication. In this reaction, multiply copies of the gene of interest synthesized in vitro by using two sets of primer. By using two sets of the primers, and these primers are chemically synthesized oligoribonucleotides complementary to the regions of the DNA, and the enzyme DNA polymerase we will use here. The enzyme extend to the primer using to the nucleotide provided into the reaction and the genomic DNA as a template. If the process of just keep the things steps: number 1 denaturation, number 2 annealing, number 3 extension. 3 basic steps. In denaturation step, both of the strands will separate by the heat. In annealing, primer will add at the corresponding ends. And extension, primer will further polymerize deoxyribonucleotide. So in this extension state, there is the use of DNA polymerase enzyme. This DNA polymerase, the use of the thermostable DNA polymerase is very important. It is isolated from a bacteria called Thermus aquaticus, so-called Taq polymerase. This is the bacteria from which this Taq polymerase was isolated, and it can tolerate even 94 degrees centigrade temperature. That is why PCR made possible. So in PCR, what we are doing? Multiplication of the gene of interest by using two sets of the primer. 3 steps: denaturation, annealing, extension. DNA polymerase used is thermostable, well known is Taq polymerase obtained from the Thermus aquaticus bacteria. Insertion of the recombinant DNA into the host cell. Recipient cells, after making them competent to receive, take up the DNA present into the surrounding. So if recombinant DNA bearing gene for the resistance to an antibiotic is transferred into the E. coli cell, the host cell become transformed into the ampicillin resistant cell. If we are spread these transformed cells into the agar plates containing ampicillin, only transformant will survive and others will die. Since due to the ampicillin resistant gene, one is able to select transformed cells into the presence of ampicillin. The ampicillin resistant gene in this case is called selectable marker. When you insert a piece of alien DNA into the cloning vector and transfer it into the bacteria, plant or animal cell, the alien DNA get multiplied into the almost all recombinant technologies. The ultimate aim is to produce a desired protein. Hence, there is a need for the recombinant DNA to be expressed. The foreign gene get expressed under the appropriate condition. The expression of foreign gene into the host cell involves understanding many technical details. After having cloned the gene of interest, having optimized condition to induce the expression of the target protein, one has to consider producing it on the large scale. If any protein coding gene is expressed into the heterologous host, the protein product called recombinant protein. The cells harboring to the cloned gene of interest may be grown in a small scale in the laboratory or maybe at the industrial scale. The cells can be multiplied into the continuous culture system, wherein the used medium is drained out from the one side while the fresh medium is added from the other side to maintain the cells into their physiologically most active, means the log or exponential phase. This type of culture method produces a large biomass leading to the higher yield of desired proteins. Small volume of the culture can not yield appreciate quantities of the product. So to produce large quantities, the development of bioreactors where the large volumes of the culture can be processed was required. Thus, the bioreactors can be thought of as a vessel in which raw materials are biologically converted into the specific products. A bioreactor provide what? A bioreactor provide optimum conditions for the achieving the desired protein product by providing what? optimum growth conditions means optimum temperature, optimum pH, optimum substrate, optimum salt, optimum vitamins, optimum oxygen, etc. In this diagram, two types of bioreactors, simple stirred tank bioreactor and sparged stirred tank bioreactor are mentioned. Yani both are stirred bioreactors. This provision of agitation is known as stirrer. Here there is no provision of spargers, here there is a provision of spargers. Spargers are meant for what? For the delivery of oxygen. A stirred tank bioreactor is usually cylindrical or with a curved base. Why? To facilitate the mixing of reactor content. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that the bioreactor has an agitator system for mixing, an oxygen delivery system, a foam controlling system, a temperature controlling system, a pH controlling system, sampling port so that small volume of the culture can be withdrawn periodically. Then all the steps after the production of recombinant protein to before marketing, all steps comes under the downstream processing. After completion of the biosynthetic stage, the product has to be subjected through a series of processes before it ready to marketing as a finished product. These processes includes what? Separation and purification of the product, which collectively referred as the downstreaming processing. The product has to be formulated with the suitable preservatives. Then such formulations has to be undergone clinical trials in case of drugs, and strict quality control test for the each product is also required. The downstream processing and the quality control testing vary from the product to product. I repeat, separation and purification, addition of the suitable preservatives, clinical trials and strict quality control test, they all come together as a downstreaming processing. In this chapter, after as the exercise you will explore, there is a one question they ask that like a genetic engineering, formation of the recombinant DNA, where in the cell cycle we observe. So you must remember, in the pectine stage of the meiosis, there is also due to the crossing over formation of recombinant DNA. Then biotechnology and its applications is also one of the part that we have to be explore. Biotechnology, as would have learned to from the previous chapter, essentially deals with the industrial scale production of biopharmaceuticals and biologicals using genetically modified microbe, fungi, bacteria, animals. The applications of the biotechnology includes therapeutics, diagnostics, genetically modified crop for agriculture, processed food, bioremediation, waste treatment, and energy production. Broadly, three parts: biotechnology in agriculture, agrobiotechnology; biotechnology in medical, biopharmaceuticals; and biotechnology for environment protection, means bioremediation. The critical research areas, three critical research areas where there the biotechnological research are going on. Number one, providing best catalyst in form of the improved organism, usually a microbe or pure enzyme. Number two, creating more optimum condition through the genetic engineering for a catalyst to act. And number three, downstream processing technologies to purify the protein should also be improvised. These are the three critical areas where their active researches are going on. Biotechnological applications, number one, here they are mentioning that there are three options that can be thought for increasing the food production. What? More use of agrochemicals, or organic farming, or genetic engineering, crop based agriculture. These are the three possible options to improve our agricultural output. Now they have their limitations. Green revolution succeeded in tripling the food supply, but not supply, but yet it was not enough to feed the growing human population. Increased yield by Green Revolution have partly been due to the use of improved crop varieties, but mainly it was due to the better management practices and use of agrochemicals, means fertilizers and pesticides, mainly due to the better management practices and use of agrochemicals. For the farmers into the developing world, agrochemicals are often too expensive, and further increases in the yield with the existing varieties are not possible using conventional breeding. So use of agrochemicals is expensive as well as it is not eco-friendly. Traditional breeding technique failed to keep pace with the demand and provide sufficient fast and efficient system for the crop improvement. So another technology is what? Tissue culture got derived. What does tissue culture means? It was learnt by the scientists during 1950s that whole plant could be generated from the explant. What is explant? Any part of the plant taken out and grown in test tube under the sterile conditions in a specific nutrient medium. This capacity to generate the whole plant is known as totipotency. Means tissue culture is based on the concept of totipotency. It is important to stress here that the nutrient medium must provide what? A source of carbon such as sucrose, inorganic salts, vitamins, amino acids and growth regulators like auxin, cytokinin, etc. A good culture medium must always provide them in appropriate proportion. By application of these methods, it is possible to achieve a propagation of a large number of plants in the very short duration of time. This method of producing thousands of plants through the tissue culture is known as micropropagation. Each of these plants will be genetically identical to the original plant from which they were originate. So they are soma clones, they are somatic clones. Many important food plants such as the tomato, banana, apple, etc. have been produced on commercial scale using these methods. Simple. Another important application of the method is the recovery of the healthy plant from the diseased plant, means meristem culture. So apical or axillary meristem are free from the viruses, hence one can remove the meristem and grow in vitro to obtain virus-free plant from the infected plants. Scientists have also been isolated single cell from the plant, and after digestion of their cell wall, they enable to isolated naked protoplast. Protoplast means plasma membrane containing cell, means cell wall devoid cell. Isolated protoplast of the two different varieties of the plant, each having a desired character, can be fused to get hybrid protoplast, which can be further grown to form the new plant, and this is called somatic hybridization. And these products, because they arise by fusion of gametes? No, by fusion of the two somatic cells. So this process called somatic hybridization and the product called somatic hybrids. Imagine a situation when a protoplast of the tomato is fused with the potato, then we know what will arise. That would be the potato plus tomato. So protoplast of the tomato is fused with the protoplast of potato, and then they are grown to form the new hybrid plants containing tomato and potato characteristics. Well, this been achieved resulting into the formation of pomato, potato plus tomato. Similarly, brinjal plus tomato, bomato. Now, is there any alternative path? Yes, genetic engineering is the alternative path. Plant, bacteria, fungi, animals whose genomes or the genes have been altered by manipulation are known as genetically modified organisms. What are the objective of these genetically modified organisms? To make crops more tolerant to the antibiotic stresses, one of the objective. To reduce our reliance on the chemical pesticides, means pest resistant crops. Third, to help to reduce the post-harvest food loss. Fourth, to increase the efficiency of the mineral usage by the plant so that the exhaust of the nutrient from the soil can minimize. And fifth, enhance the nutritional quality of the foods, for example golden rice. In addition to these uses, genetically modified crops has been used to create the tailor-made plants for the alternative resources to the industries in form of the starch, fuel and pharmaceuticals. Some applications are here highlighted. Bt cotton. Bt toxin produced by a bacterium called Bacillus thuringiensis. This Bt toxin gene has been cloned from the bacteria and then expressed into the plant to provide resistance to the insect without need of any kind of insecticide. For example, Bt cotton, Bt corn, Bt rice, Bt tomato, Bt potato, Bt soybean, etc. Bt cotton. Some strains of the Bacillus thuringiensis produces protein that kill to the certain insects such as the Lepidopterans, example tobacco budworm and armyworm, Coleopterans, example beetles, and Dipterans, example flies and mosquitoes. Bacillus thuringiensis forms protein crystals during a particular phase of the growth, and these crystals contain a toxin, insecticidal protein. Why does these toxin not kill to the bacillus? Because actually the Bt toxin present proteins exist as inactive protoxin, but once an insect ingests them, the inactive toxin protein is converted into the active form toxin due to the alkaline pH of the gut region. Consequently, they solubilize the crystals. The activated toxin now bind with the surface of the midgut epithelial cells, create their pores, causes the swelling and lysis, and eventually cause the death of the. Some Bt toxin genes were isolated from the Bacillus thuringiensis. Here the name of the gene is the cry gene. Just these are the choice of the gene depends upon the crop and the target pest. For example, cry1AC protein, cry1AC and cry2AB both will control to the cotton bollworms. Similarly, cry1AB will control to the corn borers. It means a particular gene will kill to a particular group of insects. Pest resistant plant. Several nematode parasitize a wide variety of the plant and animals, including human beings. Melodogyne incognitia infect to the root of the tobacco plant and cause a great reduction into the yield. A novel strategy was adopted to prevent this infestation was RNA interference, yani post-transcription gene silencing. I would like to tell you 5 points that you have to keep in your memory. RNA interference is a post-transcription gene silencing. RNA interference prevent translation of the messenger RNAs of corresponding genes. It is a method of cellular defense in all eukaryotic organisms. It is based on the creation of a double-stranded RNA. And the source for this double-stranded RNA means the antisense RNA may be any viral infection or any jumping gene. These are the 5 important lines. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of the specific messenger RNA due to a complementary double-stranded RNA that bind to and prevent to the translation. Prevents what? Prevent translation of the messenger RNA, yani post-transcription gene silencing. The source of this complementary RNA could be from an infection by viruses that having RNA genome, or it may be any mobile genetic element, means transposon, that replicate via RNA intermediate. These highlighted points just memorize. Not even a single question of RNA interference can ever be wrong. Using agrobacterium vector, nematode specific genes were introduced into the host plant. The introduction of the DNA was such that produced both sense and antisense RNA in the host cell. These two RNAs being complementary to each other formed a double-stranded RNA that initiated RNA interference, thus silenced the specific messenger RNA of the nematode. The consequence was that the parasite could not be survived into the transgenic host. expressing a specific interference or interfering RNA. In this manner, we obtain nematode resistance into the tobacco plant. So it was the biotechnology application procedures, both of the part that we covered. Here there I already told you that the sequence related questions or chronological questions are very significant in this chapter. So please prepare thoroughly, and specifically those highlighted areas you should revise one more time before you attempt to do the exam. Rest all things would be fine, and you will definitely score very well in this chapter also. Thank you very much, and all the very best for your upcoming exam. Thank you.